Hi, my name is Effie Nikki, and today we're going to talk about one division of the special senses, which is called eye and vision. This first slide up here, I know it's a lot of words. I'm showing you a picture of the eye. I really just want to make sure that we understand how much information we receive from our eye and how important they are. If you go back this last line right here when it says most of the eye is protected by a fat cushion of fat or a fat pad, and obviously we have our bony orbit, if you remember all the bones of the eye. We had our frontal and ethmoid and lacrimal and sphenoids in the back. So if we're take an item, we place it inside a cavity of bone, we surround it in fat, and then we're also going to see some tissue, we can kind of get the idea that this is probably a pretty important structure. That's because, bullet number one here, 70% of all your sensory information is received by the eye. So 70% of all the stimulus that you encounter on a daily basis is directed towards your eye. Nearly half of all your cerebral cortex is used to process that information. Hopefully we remember cerebral cortex, cerebrum. Cortex is that thin gray matter layer. I was going to say gray layer matter. Okay, so eye is important. Obviously we know we'd have problems if we didn't have our eyes, right? So let's look at some of the accessory structures. Most of the time we don't think about these accessory structures, and we need to just kind of go through a couple of these and see why they're important. Obviously they're going to help protect the eye and help protect eye function. The first two, pretty easy. The last three, we're going to see some pictures to help us understand what's going on. Number one is eyebrow. Thick, bushy eyebrows, as unattractive as they are there for a reason. If you're outside and you're sweating a lot, the sweat can roll down into your eye, and that could obscure your vision. So we're supposed to have nice, bushy eyebrows. Some of us choose not to, but technically they were designed for that purpose. Number two, eyelids. And you can see in this picture eyelids with eyelashes. When you close your eyelashes kind of form a little closure. Kind of reminds me of a Venus flytrap, if you know what I'm talking about. Never mind. Anyway, so the eyelashes come together and block any foreign object that might get into your eye. Also at the end, the very ridge between your eyelid and your eyelashes, you actually have tarsal glands which produce an oil to help keep your lashes nice and oiled so they don't become brittle and fall off. Those tarsal glands sometimes can become clogged with bacteria, and that's where we get a stye. If you've ever had a stye where the edge of your eye has kind of swollen up, the tarsal glands have become infected, and eventually your immune system takes care of it. But just interesting to know that's why. Okay, these last three, conjunctiva, the lacrimal apparatus, and then the extrinsic eye muscles, I have a slide for each, so let's go through those. You ever heard of conjunctivitis? Itis, by now you should know, is an infection. We're talking about conjunctiva. These are serous membrane that are going to release serous fluid and help wash and protect and keep the eye moist. So you have several here. You actually see you have a conjunctival sac, and this is what usually gets infected. Trying to get the pen working here. Here's the sac, and there's this nice little valley that bacteria and things can get into. And realize that your eye is an external surface, right? This entire section of your eye is constantly being exposed to things that are in the environment, whatever is in the air, whatever chemicals, whatever particles. Right now we have fungi and bacteria floating in the air, so these are able to come in contact with your eye. So having eyelids prevents large objects. Having this serous membrane with fluid constantly washing over your eye, that also helps. The next thing that we're going to look at is the lacrimal apparatus. The lacrimal apparatus involves lacrimal gland right here, and you can see the gland, and it has tiny little ducts that lead to your eyelid. And the sole purpose of this lacrimal gland is to wash over your eye. It produces a substance. It also has digestive enzymes in it to break apart anything, any bacteria that might be in your eye. But you're creating a fluid that washes over your eye to get rid of any particles. So if you ever feel like you have something in your eye and your eye starts watering, that's your body's way of trying to flush your eye to get whatever is in your eye out. You might notice this word, canaliculus. These are tiny little channels here on top and bottom, superior and inferiorly. We saw those in bones, canaliculi. They just mean little channels. And you can see it drains into this structure, which is your nasolacrimal duct. Nasal, lacrimal gland. So what happens if your gland is producing tons of tears and it can't get through your teeny tiny little canaliculus fast enough? You start crying. It's an overflow of your lacrimal canaliculus. You can tell people that. So also what's interesting is it drains into your nasal cavity. If you've ever seen somebody that's just sobbing, tears are coming down their eyes, and they actually will have snot bubbles coming out of their nose, it's because it's all part of this same system. So any excess is either going to come out or come down through that nasolacrimal duct. Okay, now we get to move. Oh, did I miss one? Let me go back one. No. Oh, eye muscles I wanted to talk about. I'm sorry. On this picture, just pay attention. Oh, let's try one more time. On this picture, let's just pay attention to the eye muscles here. We have several eye muscles. Hopefully when you went over cranial nerves, you talked about abducens, trochlear, ocular motor, and the cranial nerves that feed these eye muscles. So you don't have to know all the names of them. There are superior, inferior obliques and lateralis, and I don't care if you know the names. There are 6 different eye muscles that actually help us to be able to move our eye around. Okay, back to anatomy of the eye. We're going to have to break down the eye into 3 separate layers. I put what we normally call it fibrous layer, vascular layer, internal layer, and then in parentheses what sometimes you'll hear other people call it tunicas. Tunica just means layer. So tunica fibrosa, fibrous layer. Hopefully we can get that word association. All right, so let's just go through each of these, and then I'll show you the picture. So basically the sclera, this is the white part of your eye. The cornea, this is where sclera is where you're going to have muscle attachment, you're going to have nerve innervation, nerves going to the sclera. The cornea is going to be a transparent membrane. This is the first layer that light's going to pass through in your eye to get to the back of your eye where we're actually going to be able to create an image. So this is just a transparent membrane exposed to the environment, continuously being washed with the fluid produced by the conjunctiva. Okay, so now number 2, vascular layer. Hopefully we see vascular and we know something to do with blood. So the choroid layer is going to provide a blood supply. Ciliary body. These are going to help change the lens shape, and this will make sense when we get to the picture. And the iris, this is a pigment, and just make sure that we clarify this is a pigment is a protein, and proteins are made from DNA. So your iris is the part of your eye that's colored. So whether it's green or brown or blue, it came from, the reason it's that color is due to pigments, specific pigments, and those pigments are there because your DNA said make these pigments. Okay, internal layer. This is the very posterior part of the eye. This is the important part for creating an image and for you actually being able to see an object. So the retina is going to have photoreceptors in it. Hopefully you learn about photoreceptors. Photoreceptors are receptors which are proteins embedded in a membrane, and these detect light. So that's why it says photoreceptor. And then the optic nerve, this is how you get the signal to the brain. Okay, so as long as you understand the basic definitions of these, let's look at the picture. And I have them highlighted here in different colors so you can take a look. Let's start with the cornea here. So the cornea is right here. You can see that light is going to go this way. So I said this is the first object that light's going to pass through. And being a transparent membrane, it's going to refract the light in a specific way to help this image get through all of these layers. The other is the sclera. I said the white of the eye, that's all of this part back here. You're going to have muscles that are attached. You're also going to have nerves that are attached to the sclera part. So think of like a thick, fibrous outer coating of the eye. Then you can see here in red we have the choroid, which is all of this, and it runs all the way around. It's contiguous with the ciliary bodies, which are here and here. And then this is your iris. You can see the ciliary bodies have little strings that attach to the lens. The ciliary body and the iris, they're made up of muscle fibers. So when the ciliary body, when those muscles contract, it can squeeze the lens together. And when they relax, it can pull the lens apart or farther. So this shape is going to depend where the light's going to reach the back of the eye. Also the same thing with the iris here. The iris can either constrict, and this part right here is called your pupil, and we've heard about dilating our pupils or constricting our pupils to let light in. We want to let a lot of light in. Ooh, say that a bunch of times. If we want to let light in, we have to open the pupil. If we want to not let as much light in, we have to constrict the pupil. So the iris and the ciliary bodies actually help with the amount of light that's being admitted into the eye. Choroid, vascular blood supply. Alrighty. So what is vision? Bottom line. Have it highlighted here in red for you. Vision or sight is the perception of light emitted or reflected from an object in the environment. You have to have light hitting an object for you to be able to see that object. So they'll ask people, even if you were in a dark room, would you be able to see that the apple in front of you is red? No, you wouldn't. If you are completely in a box with absolutely no light, you cannot see anything. Some people say, oh, well, I can see fine when it's dark outside. That's because there's stars and there's street lights and there's the moon if you're out in the middle of the woods. There's always a little bit of light coming from some external source. Okay, light must cause a photochemical reaction in order to produce a nerve signal that our brain can notice. Blah, 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 right? Oh, look at me go. Okay, so here's light. The light is going to hit a receptor, and specifically something we call a photoreceptor. That photoreceptor is going to generate an action potential, and then that action potential is going to be carried by a neuron to our brain. So light has to cause a chemical reaction, an AP, to be created so that it can be carried to the brain. We're going to break this up into two components, optical components and neural components. So this first one here is optical components. What actually lets light get into your eye? Not how does light get changed into an action potential, that's next. This is how do we get light into the eye? And hopefully I'll remember to turn the pen on this time. Cornea. So here's light. I'm going to draw a little squiggly line. Here's the cornea. That's the first layer of your eye that light passes through. The next is in here, there's a gel-like substance called aqueous humor. Maybe it's a little bit more watery than gel-like, but this is constantly being replenished and cleaning out any waste products. Remember, we have muscles here, so in the ciliary body and the iris, if you have muscles, you're going to be generating some sort of waste from their activity. You have to be able to flush that out. And then we also have blood vessels in here, because remember the choroid is the vascular layer. So aqueous humor's next. Then the next part that it's going to hit is the lens right here. Then eventually we're going to travel past the lens into what's called the vitreous humor. The vitreous humor is a little bit thicker. It's probably more jelly-like. This fluid that's all in here is actually helping to keep your eye in its correct shape. So this fluid is actually putting pressure on these layers and keeping them in the correct shape. Okay, neural components now. These are the components that are actually going to turn light into an action potential. So we have the retina and we have the optic nerve. So in this back part here in yellow, this is the retina. This right here is the optic nerve. So what's going to happen? Light's going to come in here. It's going to do what it needs to do, which we'll look at in just a second. And then the action potential is going to travel on that optic nerve. So these neural components are the actual structures that are going to turn light into action potentials. So formation of an image, I would make sure that you do understand this flow through. This is why I've put it in again. Make sure that you can do a flow chart for this. So light's going to come in. First it's going to go through the cornea, then it's going to go through the aqueous humor, then it's going to go through the lens, then it's going to go through the vitreous humor, and eventually to the retina, where it's going to be turned into an action potential and sent along the optic nerve to your brain to register, I just saw this room, this computer. This is a blow-up of the picture that we just saw. So back here, this would be your vitreous humor. This is the layer of your retina. This is your sclera, right? All right, so we're trying to show you, guess what's in here? Neurons. So we're going to have to blow this picture up again so that we can see exactly what's going on in this tiny section. And here we have it. These are our photoreceptors. So we have, here we have rods, and here we have cones, and it's based on their shape. You can see the difference between the cones and the rods. We'll talk about what they do. One's used for day vision, one's used for night vision, and we'll talk about those in just a second. You can see here cells. How do I know they're cells? They have a nucleus. So these are supporting cells, and these cells are going to help the photoreceptors do their job. So basically, what am I saying? Light comes in this way. It's traveled through our cornea, our aqueous humor, our lens, our vitreous humor. We're now back at the retina. Excuse me. So, sorry. So light's coming in through this area. It has to pass through these other neurons and then eventually get to the photoreceptors. So only rods and cones are the photoreceptors. The others are just neurons that they're going to synapse with to help get the signal to the brain. So the light has to travel through all of these neurons to finally get to the rods and the cones. Inside the rods and cones are little pigments. And remember, pigments are proteins, and these came from DNA, right? Okay, so these discs of pigments, when light hits them, they change shape. Let's just say they go from this to this. So they change shape, and that triggers an action potential. What's interesting is these cells help make those pigments. It's a cell, it's got a nucleus, it's got DNA, it can make proteins, it makes these disc pigments and puts them in the photoreceptors. Another thing that's interesting is that you need vitamin A to help these photoreceptors do their job. So when somebody says eat tons of carrots, it's good for your eyesight, they're right. Carrots have lots of vitamin A, and you need vitamin A in order for your photoreceptors to work properly. So they weren't lying. Your grandmother, your mother. Okay, so let's go, let's see if I want to say anything else. I guess I should clear up. Once this happens, once we have, let me clear everything. Once the light comes in, changes the pigmented disc inside the rod or the cone, that causes an action potential to occur. And what's that action potential going to do? It's going to travel from the photoreceptor to this next cell, which is called a bipolar cell or bipolar neuron, and then again to these ganglion cells. They're also neurons, and you can see they all converge right here. This is part of the retina still. This whole section is the retina, but this is part of it. And then eventually these are going to travel on the optic nerve to the brain. Sounds pretty simple, huh? Okay, so make sure that you can take this picture and do the entire flow through. I often tell my students, if you want to copy this picture from the book, white out all of these words and fill everything in until you know what you're doing. That would be a really good idea. I know it's the end of the semester and we don't want to work that hard, but it really will pay off. All right, cornea here, light out here, travels through the cornea, through the aqueous humor, through the lens, through the vitreous humor, goes back to the retina, to the photoreceptor cells, right? It triggers those pigments to change shape. That creates an action potential. It travels through those bipolar cells and those ganglion cells, and then eventually the signal travels to the optic nerve and out to the brain. And we're going to look at where specifically in the brain. Some of these words here I have highlighted just to make sure that we understand some of the finer points. It's not, to me, this is the major part that you need to understand, but these might come up again, so I want to make sure. We know optic nerve, we know retina. There's something called a blind spot. Now think about this. If light is traveling here, what happens if light hits right here in this blind spot? There are no photoreceptors in that exact spot. It's almost like the channel that leads to your brain, but there are no photoreceptors there. So if light happens to reflect off of an image and hit the blind spot, you can't see it because the photoreceptors can't generate an action potential. So what's interesting, I didn't put the website up here, there's some games that you can go if you type in games for blind spots. Normally when we're looking at something, we'll have the right side and the left side, and if the image hits the blind spot, there'll be a tiny little piece missing. What's really interesting that our brain does is it copies the tiny little segments on either side and flips them so that that blind spot is filled in. So there's an interesting game where it's like two blue bars and you have to look at it for a certain period of time and do something, I can't remember. And then when you look at it again, it'll be completely filled in. So your brain doesn't want to go around with little pixels missing all over the place. So it just fills those in from the image that it sees on the right and left-hand side so that you, again, you're not walking around with blind spots. The other thing is the fovea centralis. This spot is called the macula lutea, and inside of it is what we call the fovea centralis. This is where fine detailed images are being relayed. So if light comes in and hits that exact point, you're getting to the photoreceptors without having to go through that thick layer of retina. You don't have to go through the ganglion, you don't have to go through the bipolar. It's almost like a wedge has been driven out of the retina, and at the very back you just have the photoreceptors. So light has direct access to those photoreceptors. So this is where we see fine detailed images. Okay, so now we're going to talk about duplicity theory. Why do we have both rods and cones? So one cell is used for day vision and one type of cell is used for night vision, and some people thought, why do we have both of those? I'm not going to get into the evolutionary aspect of it. We have two different kinds. We have rods, we have cones. So if we have rods, they're for night vision. Cones are for daytime vision. And this is because we have a different idea of what we want to see during that time. So rods, we're going to act a certain way, and cones are going to act a certain way depending whether we're talking about day or night. Okay, so let's talk about duplicity theory. Why do we have both rods and cones? A single type of receptor can't do everything that we want it to do. It can't do high sensitivity and high resolution at the same time. So we have night vision, we have rods. Daytime vision, we have cones. And so You guys are just screwing me up. Okay, so duplicity theory. Why do we have both rods and cones? Basically, the statement here says it all. We can't do both with the same type of photoreceptor. So we can't do high sensitivity and high resolution with just one. So we have two. Here, let me get the pen going. We have night vision, we have rods. Daytime vision, we have cones. So they each have a specific job, and let's look at exactly what those are. You don't need to memorize everything that's on here. I'm just trying to help you understand why we have two different types. So in this picture, we're showing you the rods here. So this is nighttime vision, right? You can see that there are several rods that converge on a bipolar cell. So normally this picture doesn't do it justice. There's about 600 rods that converge on one bipolar cell. So this is not really specific. This is why we have a great deal of spatial summation. You can see a lot, but you can't resolve the detail. I like to call this fuzzy. You're out at night, you can see something in the shadows, but you don't quite know what it is. It's just kind of fuzzy. That's night vision. Its sole purpose is really just to alert us to motion. Do you see something coming at you? Can you do something about it? The next one is day vision. And day vision here, we can see cones, and for the way that they're shaped. And you can see that each cone has its own private line, its own bipolar cell, its own ganglion cell, and it carries it to the brain individually. So these are it says no neural convergence, not many converging on one. They each have their own private line to the brain. This is high resolution vision, but little spatial summation. So you can see everything individually very, very clear. You just can't see a whole bunch. And that sounds a little untrue. You can see farther when you have night vision. It's just not as fine-tuned. During the day, you're looking at each individual thing very precisely. You just can't get in as much other information, almost because like you're overloaded with all the fine detailed information. Okay, these last couple of slides are really just for me. I don't know anybody that really tests on these. Again, you can ask your instructor. I just want you to see, why do we have glasses? What does nearsighted mean? What does farsighted mean? So this is showing you an image. The big R here, you're looking at the letter R. And these are what we call focal points. So what did we say? Light refracted off of an object, and that's going to travel through the cornea, through the aqueous humor, through the lens, through the vitreous humor, to the retina or the back of the eye. Photoreceptors are going to do their job, but the image is inverted. And eventually, they're going to send their signal to the optic nerve. The action potential that's created is going to be sent via the optic nerve to the brain. So in distant vision, you can see that the object is pretty far apart, and your lens, the way that it's shaped, these ciliary muscles are contracting and pulling your lens tighter. In close vision, what's happened? What do you notice about the lens shape? These have relaxed, and now your lens is kind of fatter. So instead of elongated, it's kind of smushed together a little bit more. So then the image again is reflected or refracted, inverted on the back of your retina. So what happens if you're nearsighted or farsighted? All right, so nearsighted, it tells you what you are. Nearsighted, you can see near. So I say no far. You can see near, but you can't see far. And if you're farsighted, you say no near. You can see far away, but you can't see close up. So the best way to explain this is, depending on where the image is located, whether you're looking close or far, the shape of the lens, where the image ends up being inverted, where the image stops, will determine whether you're nearsighted or farsighted. So what happens if your image is inverted right here? Can that image get to the photoreceptors to create an action potential to travel on the optic nerve to go to the brain? No, it can't. So this is an example of you can't see really far away. Your image is moved forward. If you're farsighted, your image is moved too far back. If you're outside of the retina, you're not going to be able to see that image either. So what do? We put a lens in front of it. And then as the light travels through the lens, it's distorted by the time it hits the your actual lens in your eye, and then it can move this image. So when you go to the doctor's office and they're putting those little lenses in front of your eye and they're making you read the board and say, does this look better or does this one look better? And they go through that back and forth. They're trying to find the exact right curvature of lens that's going to put this image exactly where it needs to be in order for you to see. Pretty cool, huh? Okay. This is just interesting for me, so that's why I put it in here. I want us to understand some of the things that we've talked about in coordination with the brain. Remember, 70% of all of our visual sensory information travels to the brain. Half of our cerebral cortex is used to process this information. So what I want to show you here is the right eye versus the left eye. So here is your right eye. Your right eye can see all of this. So this is right. What the color deviation is trying to show you is this information travels on these blue pathways. This information travels on the yellow pathways. So what's interesting is even if you were missing an eye, you still have quite a large vision scope, if you will. Either eye, it doesn't matter. They overlap. It's not like we only see this 90-degree angle. We have a much wider angle with each eye. What's interesting is where it deposits this information. So again, when I'm showing you these pathways in the brain, they are neurons, right? So information from your right eye, it stops here, which is part of your hypothalamus, and then it stops here, which is part of your thalamus. They don't have hypothalamus over here, but these are areas within the hypothalamus. Let's see if I can spell. Okay, so do you remember talking about the hypothalamus and saying it's a timekeeping? It has a timekeeping function. How do you know how long you've been asleep, how long you've been awake? We usually have visual cues, the sun, right, the moon. So part of the information of your vision goes to your hypothalamus so that you can keep track of your day. Another part goes to your thalamus, and this has something to do with reflexes. So if the light is shining too bright in your eye, that information goes directly to the thalamus and you have a reflex and it causes you to close your eye or constrict your pupil or the iris, the muscles around your pupil. So, oh, I'm sorry, this is the last part I wanted to talk about. The occipital lobe. Eventually that's where the information ends up because your occipital lobe is that visual cortex center where your brain decides or recognizes or has conscious thought about what you've just seen. So again, I don't think you're going to be tested on the visual pathway of the brain. I just think it's interesting that we have functions like reflexes and timekeeping, and we have to tie in. When we're learning something new, we got to tie it in with what we just learned. Well, I hope you enjoyed this lecture, and I'll see you next lecture. Bye.